So we analyzed cancer cell line and identified this network. So these genes, upregulated genes, are controlled by these master regulators. So when we look at that network more in details, we found there are proteasome and cell cycle components which could be very good targets for various drugs. And finally, we identified these drugs which are most perspective for treating this cancer sample. Hello everybody. You are at the channel My-Genome-Enhancer. Today we'll talk about analysis of data from lung cancer. We'll have actually genomic data, sequencing of the whole genome, and the transcriptomic data, and actually many other types of data. You might be interested and might be wondering where you can get so many data about one sample, about one cancer sample. Well, basically, I must say that's not really a sample from a particular patient, the fresh one. It's a cell line. So we get it from Cancer Cell Line Encyclopedia, and that's the cell line called NCIH 1975, which is actually was isolated from a lung of a non-smoking female with a non-small lung cancer. Basically, this is a true sample from one patient. And we are going to analyze it. So as I said, the data we took from the Cancer Cell Line Encyclopedia, we'll see that this cell line is characterized by many different types of assays. Basically, we have a copy number, expression data, all kind of data about mutations, about fusions, even proteomics. And of course, this cell line was actually subject of scanning through different drug sensitivity assays to many different anti-cancer drugs. So we will have this data. But actually, we will treat it first as we would have no such amount of data. So we will take just expression data and the data about the mutations in the whole genome, and we'll try to analyze it. And the tool we are going to use, as usual in our channel, we are going to use the Genome-Enhancer. So let's first have a look at this data, which we actually downloaded. So first set of data is actually the expression data. So it's present here in Excel file. So this is a gene expression data, RNA-seq, for many cancer cell lines, and we selected particularly for lung cancer cell lines. And our cell line is inside this data, so one of the column. And as you can see here, we have read counts which were measured by RNA-seq. The second file is actually this VCF file from this cell line, and as you can see, this is information about substitutions, mutations, which were observed in that somatic genome of that cancer cell line. Okay, let's log in into Genome-Enhancer using our standard login. Here we are, and the first thing we do is to start a new project. So let's call it lung cancer NCIH 1975. So the project is ready to upload files. So first, as you remember, we have the files with genomic mutations. So this VCF file. So let's load this file. So we click here. We find this file in the folders. Here it is. So it's uploaded, and now we are going to import it. So of course it defines the VCF format. And genome build is HG19. It's detected automatically because in the file there is the HG19 is mentioned. If this file is not, if the genome build is not mentioned in the file, then you have to basically define the genome build which you should know. So we click import. So the file is imported. So as for the gene expression data, we first have to prepare this data. So as you remember, we had this Excel file with all the lung cancer cell lines, but now we are interested just in one cell line, and we get this data from that file. But what should be the control? As a control, we decided to look at the normal lung tissue, and expression data of normal tissues are stored in this database called GTEx portal. So you can get a particular slice. So you get the data about the normal lung. So here you see this data about normal lung. So we have to download this data, and finally we look at that, and this is also read counts. And we put it in one Excel file. So here you see this Excel file where we have ensembled gene symbols and gene counts for different samples from normal lung. So you see how many there are in this database. And the first column in this table is actually the read counts from the lung cancer sample from this NCI H1975. So now we have to load this data into our platform, basically in the Genome-Enhancer. So let's do that. So we find this data. Let's load it. So the file is quite big. It takes a bit of time to upload it. So upload is ready. Now we are going to import it. So first it is detecting the format. So the format is detected. It's a tabular format. And the data type is transcriptomics. It's also found where the data start from the second row. And the first column is presenting the ensemble IDs. So everything is fine. So now we are importing it into the platform. So the data uploading is ready. So let's have a look. But first, before we actually start the analysis, let's us further prepare the data. So for that, we'll go to platform. So the button is somewhere here. Here it is. And here we are. Basically, you see this platform view, and now we can go to this project and look at the data which were uploaded. So this is a table which we have uploaded here. So we have here the read counts for the cancer cell line and read counts for the samples of the normal lung. So let's calculate the log-fold change and find differentially expressed genes. But for that, we have to normalize this data because they came from different resources. And for that, we will use the method of normalization, which is called DSEQ. So it will normalize the data and also will calculate differentially expressed genes. So let's start this method. And now let's put there this table. And we have to define the samples which we compare. So here the experiment, so the sample which we are going to analyze in our focus is the sample from lung cancer. So this sample. And the background, which are control samples, are all the other samples. So we will take the same table and select all other samples as a control. And we have to specify the output. So this will be the calculating the log fold change. And let's run the analysis. So we got this table of fold changes. You see there? So we have the column for log fold change and for p-values and for adjusted p-values. We can even sort by these columns. And you can see here at the top those genes which are mostly differentially expressed. So here, let's use this filtering in our platform. And let's filter out those genes which have no expression at all or very little expression. So for that, we filter above threshold, and let's filter like base mean should be bigger than 1. So this is 21,000 genes which are expressed, at least on a low level, in the cell line, and also have calculated log fold change. So let's save this as a separate file. Filtered mean value 1. Good. So next is to specify this table as a particular omics type. This is a transcriptomics type. So we set it as transcriptomics. So basically then algorithm will know what to do with that data. And now let's return back to Genome-Enhancer view. So we go back to the wizard of Genome-Enhancer and make a next step. So for the next step, we need these files. So first of all, the file with mutations, the VCF file. And now we need this file with log fold changes. So it detects immediately that there are several columns in that file, and we can select each of that column as a main column for our analysis. So we press next. And now it's actually have prepared for us experiment and control. So it would be able to do this comparison by itself. But since we have already done the calculation of log fold changes, so we basically don't need the control here. So it is already compared to the control. We just name this accordingly. So basically, this is NCI H5 transcriptomics and mutations. So and now we put here our mutations, and we put here the log fold change. So it will basically take the whole list of genes and will select the upregulated and downregulated genes from that list, and also will select non-changed genes in the middle to compare with in our further analysis. So we press next step. And now we have to select the disease. So first, let's select the general term, lung neoplasm, actually, lung neoplasm. So let's add also non-small cell lung cancer, carcinoma. And now we have to select this category of transcriptomic and mutations. It actually detects that this data contains genomic data, so data about mutations. So we also can generate MTB report, which is called molecular tumor board report, a report about the influence of those mutations and known mutations and actionable mutations in the clinic. So we select that to do. And so we select this, condition 1, and we have just one condition here, and then we start the analysis. So now the analysis is started. It will take several hours, so we can have our lunch in between, and then we'll see the result. See you later. Today I will eat tomatoes from our garden. So here is a full report of the results of the analysis. So the program formulated the main finding so that these two genes are actually found as a promising druggable targets to treat this particular sample. The activity of those genes are controlled through these transcription factors, E2F1, SRBP, F2, and androgen receptor. So let's have a look at the results. So first of all, in the data section, we see that indeed we took transcriptomics data and data from genomics. So basically those mutations in the genes and in whole genome. And first, what the program is doing, it's taking this transcriptomics data and classifying genes to upregulated and downregulated. And also it selects so-called non-changed genes, which are in the middle of that scale. So, and the top upregulated genes are here, and top downregulated genes are here, so you see log-fold change. So then those up and downregulated genes, basically the program took 300 top highly upregulated and top downregulated genes. It maps it to, first of all, to gene ontology. And this is a result of the gene ontology mapping. So you can see those big categories which are enriched among the upregulated genes. So what you can see here is mitotic cell cycle progress, regulation of mitotic nuclear division, regulation of chromosome segregation, cell cycle checkpoints, and so on. So cell division, so those categories which are clearly defining that these cells are rapidly proliferating, and that's typical picture of the tumor cells. So then those genes are mapped to various pathways, signal transduction pathways, and what you can see here, pathways of cyclosome regulatory network, in general cyclosome regulation, G2M phase transition, Aurora B cell cycle regulation, so some small signal transduction pathways leading to cyclin B, and in general metaphase to anaphase transition. So indeed, clearly you can see those pathways which characterize the rapidly proliferating cells. Then those genes, upregulated genes, are mapped to the human diseases. So basically, these are the various biomarkers of human diseases. So those 300 genes of upregulated genes are mapped to various biomarkers of many different diseases. And here you see top 10 found diseases. So among them you can see neoplasms in general, especially epithelial, breast diseases, breast neoplasm, carcinoma, adenocarcinoma. So there is no specific lung cancer featured here, but generally carcinoma, breast neoplasms are featured here. So the same is done for downregulated genes, and this actually shows a different picture what is downregulated in the cells. And what you see here is immune response, humoral immune response, leukocyte migration, again immune response, immunoglobulin production. So a lot of functions which are related to immune response, which has to be suppressed here in those cells. And when we put this on pathways, you see the particular pathways which are downregulated. And in terms of diseases, clearly these genes which are related to immune response and which are downregulated here belong to as biomarkers of autoimmune diseases, infectious, arthritis, and things are related to immune system. So So in summary, you see that in terms of gene ontology, what is upregulated is chromosome segregation, cell division, and what is downregulated is humoral response, phagocytosis, by the way. So the next step which this program performs is actually searching for the regions in the genome which are potentially affected in those cells, so which may play a role of specific enhancers which enhance the regulation of the genes in these cell lines. So, and how we first of all look for the enhancers. So the general concept is that those mutations which are observed in that cell line, of course, a lot of them are just random, but if you look at them carefully, we may see that some of those mutations are the subject of a certain selection in the cell line. So those mutations which are destroying or actually creating some regulatory modules or regulatory sites could be subject of special selection in those tumor cells. First of all, we took these all mutations which were detected in that cell line and mapped them on the various genes. And here in this table, you see the list of top genes with many mutations in them. Here you see the blue bars correspond to the exons of those genes, and of course, the longer the gene, the more you get mutations in it, and the red bars are the mutations. And here the number of mutations which were observed inside those genes, including 5' and 3' regions of those genes. Next is actually we are checking if those mutations which are observed in that cell line in comparison to the reference genome, they are either they are creating new sites or destroying the existing sites for transcription factors, sites for binding of transcription factors. And we also calculate here some statistics, basically, and based on the number of mutations which leads to the gain of the site of a particular transcription factor motif or the loss of the sites. We can compute the probability of those so many losses just if they happen by random chance. For instance, if you take this first motif for CNP8 factor, so you see that in terms of gaining 13 times we observe the gain of that site. So in reference genome there is no site, and upon mutation this site is actually created. And then only 13 sites. But there are 2,693 sites which were lost. And if we compute the p-value, we can see that this number of sites which are lost is much more than we could expect by random chance. So the same as for other factors. Sometimes you get a very high p-value for the loss, like NF-kappa B, for instance, also p in minus 10. But sometimes you get a good p-value for the gain, as well as for the loss. For instance, E2F3, so the transcription factor of E2F family, many times it is created and many times it is lost in various positions. So, which leads to the idea that this gain and loss of sites can have a functional role in that genome. So, so other factors, some of the factors, for instance, SP1 is definitely, we get a lot of gains, but very few losses of those sites. So from that analysis, we can estimate which transcription factors are important in that genome to regulate various genes in that genome. So the next step is to analyze the whole regions which are inside those genes, which are differentially expressed and which are flanking the most important enhancers. So actually how we define them. First of all, we take the upregulated genes and downregulated genes, and then we look which genes contain those mutations. And if there are regions in the regulatory regions of those genes, so in introns, in 5' regions, or even in 3' regions of those genes, which have a lot of mutations, look like clusters of mutations, we define those regions as potential enhancers. So because they are located in the regulatory regions of the upregulated genes on one side, and on another side we see a cluster of observed mutations, which actually changing transcription factor binding sites. We focus our attention on those regions as potential enhancers. So the length of those regions is around 1,000 nucleotides. And then we apply the composite module analyst, this program. You can read about this in the comments under the video. It searches for the composition of transcription factor binding motifs, which are describing those regions all together. And it found it takes several hours to optimize and to find these combinations. We apply here genetic algorithms which are working to find optimal set of motifs and optimal parameters of those motifs, such as cutoffs and number of sites which are taken into account. So more details, you can read it in our documentation. So, and then these combinations of motifs, they discriminate those regions of our focus from other random regions in genome. And on this slide, you see that the red bar, the red bars, they describe the score of this composite module among the regions of our interest, and the blue bars describe these scores in the random regions. And the discrimination is significant by Wilkinson p-value test, and actually AUC value is 0.77. So it's significantly discriminating from each other. It's not the best discrimination which you can observe sometimes, but actually statistically significant. And it gives us an idea which transcription factors here they work together to regulate those genes. So among the motifs which we found here, which are building this composite module, we can see a motif for E2F factors. Actually, we see it 3 times here in this half of in the first module of the whole complete model, and here 2 times. So definitely E2F factors are indeed very important here. Then we see here GATA factors and FOS, June, and also the OCT family factors and FOX family factors, and some other important factors which definitely playing a role in cancer development, in differentiation. And actually very interesting is NANOG factor, which is involved in stem cellness properties. So the same is done. First of all, we can have a look at those genes which are regulated by these factors. So we see them in this table. So they are sorted by this CMA score, the composite module score. So at the top, we see those genes which are found here in this histogram. So and the top one is transportin, for instance, and you see other genes. We can have a look at those genes in more details in order to see the positions, the actual positions of those enhancers. So you see this region, enhancer region in the genome, and which transcription factors. So we can sort by CMA score. And for instance, here in this TNP1 gene, we see in the center of that region there is a cluster of binding sites for these factors, and especially you can see a lot of factors for E2F, a lot of sites for the E2F factors and other factors which are playing an important role here. So and you see the same cluster, similar clusters in other genes. Okay, so we'll go back to the report. The same is done for the downregulated genes. And here you see partially similar transcription factors like FOS and FOXO and relay like NF-kappa B, but also OCT factors. But for instance, we don't see here E2F factors, so which means they playing a role in upregulation of the genes, but not so much in downregulation, perhaps. And again, we see this discrimination of the enhancer regions of the downregulated genes from the random regions in genome, and it has a similar AUC value and statistically significant discrimination. And again, you see those genes which contains those enhancers in the regions, and they are sorted here by this composite module score. So finally, if we look at these transcription factors, so taking together those analysis of the transcription factor motifs which were found in the composition in the enhancer regions, and also transcription factors whose sites are affected by the mutations and whether these sites are created or lost. So putting all this information together, we see these are the most important factors which this program is found in the analyzed genomic regions. And here we see the regulatory score, which tells us how important this factor in the regulation of the genes, and also the yes/no ratio, it telling how significant is enrichment of the motifs of that factor, just individual factor, in these enhancer regions which we are analyzing. And sometimes you see this enrichment like 10-fold, and in the case of E2F1, it's 4.3-fold, so pretty significant enrichment of the motifs in those regions. And the same we see for the downregulated genes, so the transcription factors which playing a role in the downregulations of the genes. So the next step is to look whether those mutations can influence on the signal transduction network in general. So basically, some of those mutations which happening in the coding regions of genes, they are so bad, so they, for instance, some of the mutations, they even lead to of gaining of a stop codon or a frame shift, and in such a case, this protein is probably not functional anymore. So in such a case, we list those proteins, which you see here, which are destroyed, sort of, by those mutations, and then we exclude those proteins from the signal transduction network, which we then analyzing on the next step. So, and analysis of signal transduction network leads us to identification of these master regulators. So in the upregulated genes, so master regulators which potentially involved in regulation of upregulated genes, and for downregulated genes. So this is a table, but of course it's much better to look in the graphical form. And finally, this is our network which the system identified for the upregulated genes. And let's have a look in more details in this network. What do we see here? So we can click here to see the full diagram. So as you can see, there are interesting proteins which are pink here, and let me explain what does it mean. So, but first let's have a look at the bottom of the picture. So here at the bottom of the picture you see the genes. So these are the genes which are upregulated first of all, and then which actually contain mutations. So in the regulatory regions of those genes, in upstream or or 3' in 5' or in 3' regions or in introns of those genes, we observe clusters of mutations. So these are candidates of the genes which are actually playing an important role in the pathology of those cells. And among them, you see, of course, several genes which are very interesting, like BRCA1, EGFR, and other important genes. At the top of that picture, you see the proteins which are actually encoded from that genes. For instance, BRCA1 is encoding this BRCA1 protein, right? And then this picture has a kind of a circle inside. So as you can see, there's BRCA1 through a signal transduction cascade leads to the activation of this set of transcription factors, which are blue here. So these guys. And then these transcription factors, they are combined in these composite modules, and they in turn are regulating activity of those genes. So for instance, BRCA1 gene activity is regulated by these factors. As a result, the BRCA1 expressing the BRCA1 protein, which in turn lead to the activation of those factors in a second place. So basically, we see here this kind of positive feedback loops, and those dashed lines here are indicating these positive feedback loops. So all of these pink proteins here, which we call master regulators, they are encoded by these genes. And these master regulators, which you can see here, are the candidates for as a targets for therapy. So then we return back to report. And as you can see here, we also construct the network for the downregulated genes. And you see here different master regulators which are involved in downregulation. Some of them are the same, but some are different. And then among those master regulators, we look more in details and try to identify the important potential drug targets. So as you know that not all proteins in the cell are potential drug targets. Some of them are so deep in the cell it's very difficult to target it. Some of them are on, like receptors or more accessible for treating by various means. And first of all, we will look at those proteins which are already known as potential targets. And here you see the list of those proteins, and here we have this druggability score, which basically the number of known drugs which are targeting this protein in various diseases. So already those drugs which are already on the market. And among them, for instance, you see this polo-like kinase 4 has actually 28 drugs which are on the market, or proteasome inhibitors, here you have 2. And we are actually ranking those drug targets from the point of view of their role in that network as master regulators, and also from the point of view of druggability. So we are balancing these two approaches, and then from this balance we calculate this total rank, which allows us to rank the master regulators, to rank the targets, and give priority to those targets which on one side are important master regulators in this cell line, on another side they potentially druggable. And the same we can see for potential druggability from a point of view of chemoinformatics. And that's another point. Actually, here we see the druggability score built on the basis of known drugs, but here we take a big collection of active compounds, not necessarily drugs, and we compute using chemoinformatic program pass the druggability score of each of that protein from the point of view of the calculation, chemoinformatic calculation. And here you see some other targets which may be important here. For instance, you see here the various sub-units of proteasome. And it's appeared to be that proteasome is indeed a very important target here. So the system picked up very few targets, which we can see from many various points of view. And at the top, you see here actually proteasome, also cyclin B and CDK1. and other proteins which are involved in that cascade. On this schema, you see that these potential targets, which in turn can be targets for these drugs, and we will talk about drugs later. They, together with other signaling molecules, they activate these transcription factors, which are in turn activating their target genes or repressing their target genes, and which belong to these GO categories, which we already discussed above. Next, we are looking at the part which is talking about identification of potential drugs. So we found potential drug targets, and these drug targets are associated with many drugs which are used in the practice, in clinical practice, or in research to target those targets. And the big collection of those drugs we have in our database, and we can associate those drugs with these targets and give a list of drugs. So that would be helpful for research and for potential use in the clinics. So here, first of all, we look at the drugs which are approved in clinical trials for this type of oncology. So again, we are talking about lung cancer, specifically about non-small cell lung cancer. And the program listed here the drugs which are associated with these targets, which are found here as potential targets in our analysis. And we are also looking at these somatic variants, which are observed in some of the targets, and which are known as actionable targets in the clinics. Some of those mutations, they can either tell us about the potential response to that drug in lung cancer for the non-small cell lung cancer patients, which is observed in many patients and even improved by FDA. And sometimes these mutations, some mutations are telling about resistance. And as you can see here, the first two drugs, which are erlotinib and gefitinib, they are ranked by our system as the top drugs, and we get almost 100% score for them because their targets are multiple targets. As you know, they are actually kinase inhibitors. They are among our targets which are found here. And but here we found that some of those mutations which are observed in that sample, they are known to provide a response to that drug or to provide resistance. So both mutations are here. So this mutation is actually lead to the response to that drug, but another mutation leads to the resistance to that drug. And that's so-called A1 evidence, which actually FDA proved and confirmed by many clinical trials. So, which tells us is some sort of contradiction, and in such a case, we would we would tell that these drugs, which have this contradictory evidence from clinical trials, they are removed from the list. So we are not really considering them further on. So as you can see, the top drug which was predicted by the system is paclitaxel. And the drug score is pretty high, it's 94%. And it is known that this drug is actually FDA approved for treating non-small cell lung cancer. And for this particular cell line, it seems this drug has the biggest potential. And indeed, when we looked at the data about this cell line and how the cell line was treated by various drugs and with various concentrations, they actually checked whether whether the cell line is sensitive or resistant to that particular drug. And in the first essay, which was published in Cancer Cell Line Encyclopedia, you can see that paclitaxel is at the top of the list, which is sorted by this ACT area. Actually, the higher the ACT area, the more sensitive is this cell line to that drug. And you see at the top, again, this paclitaxel, but there are also irinotecan and popotecan, which are a target for this topoisomerase 1. And there are other drugs, and erlotinib, for instance, here with a quite low score here, as it was predicted. Later on, these tests were repeated with many other drugs, and in a bigger list, which was published recently, we can see that, again, paclitaxel is at the top of the list. This list is actually sorted by IC50. And at the smallest, the IC50, the most sensitive is this cell line to that drug. And you see this bigger list of drugs. And again, the paclitaxel is among the top ones of that, for that cell line. What about bortezomib, which is actually at the top of that list? So this cell line is actually maximally sensitive to this drug. And actually, if you look back to our report and look at this picture, you see bortezomib is actually an inhibitor of this proteasome. And it is predicted here as a potential drug for this cell line. And we also see here in this list, when we extend it further and look in the full list of drugs, we can see this drug as well. So it has a drug score 80, which is pretty good, but of course not at the top. So in summary, by analyzing genomic data and also transcriptomic data of this cell line, NCI H1975, which is a cell line of a lung cancer sample, we identify these two main targets. First of all, actually cyclin B, CDK1 complex, and basically all the components of our regulation of cell cycle, and proteasome as a target for the drugs. And about the drugs, we see here bortezomib as targeting the proteasome, and also paclitaxel, which is at the top of the list of the FDA-approved drugs for this type of cancer.